However, back to you, Teresa. We heard while we were at ESMO here in Barcelona about the 202, the 5202 of the Insight drug. Tell us more about this study.
Yeah, this is a single-arm phase 2 trial with another FGFR2 inhibitor, the Insight drug, pemigatinib. They reported in 107 or more than 100 patients with cholangiocarcinoma and the FGFR2 fusion.
Yeah, 146 actually.
Yeah, I think it's, yeah. So response rate of 35%, I think is 35%.
Correct.
So again, in second or beyond lines. So I think this is a very an impressive results for this population. Again, the progression-free survival, it was pretty high. And the overall survival that is completely mature, it was 21 months. So I think that this opens light in this kind of patients. Incredible results for me, are really good.
Sure. No, by all means. And Martin, however, they kind of reported on 3 cohorts with the FGFR fusion, mutations or other alterations, and nothing at all. Tell us a little bit more about those 3.
Actually, it's kind of reaffirm what we have been talking about for the last hour and a half. Is the intrahepatic, the fusion is really a narrow, very tunnel view where the responses were. We find out that the mutations and obviously the ones that doesn't carry that driving mutation, the response rate were not existent. So it just reinforced what the population should be for the next study.
Fair enough. And if anything, we're happy to see that data. Actually, in full disclosure, again, many of us were involved with this kind of effort, same like for the BGJ 398. And I would say that definitely it's quite intriguing, as we just heard from Andrew, like, response rate, like more than, actually, I'm sorry, from Teresa, like response rate more than 36%. In addition to that, yes, there was kind of a delineation to see what other changes genetically that could occur that could have influenced the choice of therapy, being the fusion proteins or any genetic changes or even no genetic changes, which, by the way, does not mean no genetic changes, but it's really something that we don't yet understand or know per se. But now we know very well that actually the pemigatinib is also moving on with the phase 3 trial, again in first-line therapy versus GemCis. So quite a bit of action that's happening in that early game.
Can I ask you a question?
Yeah, please go ahead, Andrea.
So why, I guess one of the thought is, why not combination? Since we're talking about GemCis, versus a frontline target therapy, is there a thought doing the combination versus we think there's not synergy versus we think it's much toxicity?
My kind of simple answer to that, first you want to know what your asset knows, what it does by itself before you start combining it, because otherwise you don't want to piggyback on something else. And in other words, I would say presumably, yes, it might be looking into the future of that, but we need to make sure also there is a potential or no potential synergistic effect. Martin, you seem to have some thoughts on that.
Yeah, right. I also tweak on the same. I think it's better to know the activity on a single agent up front in the first line and where these studies are moving. If we learning from some of the other diseases, that when we start to combine some of the TKIs and chemotherapy, not necessarily were better. Okay, like specifically in the lung cancer area, they were actually increased toxicities and responses and survivals were not the same. So it's actually worth to just look at as a single agent up front.
Absolutely. Fair enough. Andrew, back to you. And this is something that I have to admit, it continues to intrigue me quite a bit. And it's nice work that was brought up by your group with Lipika Goyal and yourself, is that interestingly, when you gave the BGJ to certain patients, if they were to kind of progress on the therapy, you were able to delineate that there were genetic changes that occurred to stress out the tumor one way or the other. Tell us a little bit more about this finding.
Yeah, so we have learned quite a lot from target therapy development in oncology, period. We definitely know that these target agents, even though they work well, they almost invariably will develop resistance. And the resistance can be further classified by intrinsic resistance, meaning they just don't respond to the drug in the first place. Or more commonly or more meaningfully, they will actually respond to the treatment, but then because of the very significant molecular heterogeneity occurring while patient receiving the drug, the tumor actually develop the acquired resistance. As a result, that contribute to the therapeutic failure. So understanding this mechanism obviously has significant impact. We have learned from target therapy development in lung cancer, melanoma, and figuring out this mechanism really can guide further therapeutic intervention. So in the case of the BGJ FGFR inhibitor in cholangio, we were able to demonstrate as you're getting the treatment response and the tumor is actually starting to emerge with very meaningful mutations. And the specific mutations that we identified is actually in the FGFR kinase domain. So these are the mutations that actually have the theoretical consideration of altering the binding of the drug, with the emergence of the mutation. So I think the implication with the identification of these mutations is that we're able to guide these patients with additional therapeutic interventions, particularly that we have the next generation of the FGFR inhibitors, that they may actually overcome the mutation that's actually already emerging in this setting. So clearly this is something that's actually definitely doable, but also that actually has clinical implication.
Absolutely. If anything, it's quite fascinating work. I strongly recommend that you look at this manuscript by Dr. Lipika Goyal and with her senior authorship by Dr. Andrew Zhu, because it's quite fascinating. I mean, I have great admiration. They actually did really stat autopsies on patients with cholangiocarcinoma to be able to dissect the tumors from different sites in the liver and understand whether genetic changes that occurred up in progression or, of course, sadly, up in death. And if anything, yes, they were able to delineate some of them. Admittedly, we still have a lot to cover in that regard, because the question is it a haphazard you stress like a gatekeeper mutation, which is, for example, the V154C, or could it be that could be some evolution that's happening on different models that are yet to be discovered? Something that we'll wait and see where this is going to take us. With this said, however, I'll go back to Teresa, and in your perspective, in regard to the FGFR2 fusion, there are a lot of other efforts going on in that regard. Actually, it's a very busy field. Tell us a little bit more about your perception, at least, on that.
Yeah, in fact, in that moment, all our new compounds are in the same setting. So it seems complicated to understand which is the exact place for all of these new drugs, because it's only a few percentage of patients with intrahepatic cholangiocarcinoma that is a rare disease. So it seems incredible, but they are there. There's some of these compounds, for example, the TAS compound, that it seems to have some differences. For example, it seems it could work in these resistant patients previously treated with the FGFR2 and that becomes resistant. And we have some examples of patients that respond to this because there's some differences in the drug, they are irreversible. So maybe we can have these small differences. If not, they are all in the same setting in this moment.
Absolutely. And Martin, back to you on this issue of the different therapies for the FGFR, and of course the alterations that can occur based on stressors of the tumor by certain therapies. How often do you see not specifically I'm talking about the fusions, but the mutations, other alterations in regard to FGFR2?
It's probably around, I'm going to say, and feel free to correct me, it's probably around a 15%, 20% when you start to see the other mutations. What is fascinating to me is that the fusions, the partners of the fusions are so large and so variable. And I'm not sure we have really dissected that yet, if that have any different meanings depends on the partner.
I'm glad you bring this up. If anything, this is exactly what you just spoke about. There are so many alterations that are occurring out the FGFR2 fusions that, number one, where does it start? How does it get stressed out by the tumors? I think Andrew already brought up a very important point, which is what other therapies can come into play afterwards to see where things are. With this said, to wrap up the FGFR2 story, at the moment we have many products. We heard about several of them. We know that two of them at least are moving into a phase 3 trial, among which the BGJ398, which has plenty of data that as we heard, many of us have collaborated on, and we're doing it all collaboratively together. The other one is, of course, the pemfimgritinib of the Insight that we also heard about here in Barcelona yesterday with the Fight 202, which again is intriguing, especially because of the perspective, not only look at this nice high response rate of 36%, which is applicable to the fusion cohort A, but also the intriguing part in regard to the mutations as well.